Well, welcome everybody to our winter series of our Ecology and Evolution seminar series. It's great to have you all back again and to start off this winter series together. So winter series on this beautiful sunny day. We've chosen this theme for our winter series of conservation technology. And that theme's been chosen based on feedback from people that have been involved in our autumn series. So during autumn, we were doing a bit of a trial and we asked for feedback from people and did an online poll. And thank you to everybody who responded. We had 50 responses. We had polling on some suggested titles and also had an open invitation for other suggestions to be put forward. So we've got some great ideas for some series coming up, and overwhelmingly the biggest support was for conservation technology. So this is a really exciting cutting edge area of conservation that everybody's really keen to hear more about. So we've chosen conservation technology as our theme for this winter. And a shout out to Megan Lewis for her gorgeous photo as our hero image for this winter series of conservation technology. And next month, we're going to be hearing from Megan and we'll be able to hear a bit about how this image came about and where is it. So I'd love to see in the chat line, put in your guesses of what do you think? Where was this photo taken?
Yep, okay. Is it a photo?
Yes, yeah, absolutely right. I actually meant image. So where is this image? So there you go. Yep, Megan is giving you some clues.
To be revealed.
Yes, love to see in the chats your thoughts on where this is from. And for today, so that our kickoff for our conservation technology. So we've got a bit of a theme around conservation technology and also some other aspects. So just a reminder that for our PhD seminars, sometimes they align with our series theme and sometimes they don't. They take us off into other exciting areas of research. So today we've got a bit of an overlap with our theme. And we've got two really exciting presentations by two of our fabulous PhD candidates. So Brittany Williams and Vittoria Cipriani, who are doing some really exciting work in complementary areas. So, and I've loved the bios that they put together and their stories about their research. So really excited to hear more about that. So we're going to start off with Brittany Williams, who obviously does not like oysters, but is out there doing some great work with her supervisors, Connell and Dominic McAfee, around oyster reefs.
Okay, so good afternoon, everyone. I'm Brittany and I'm in the first year of my PhD, and my topic is on enriching marine soundscapes to restore Australia's lost native oyster reefs. And my supervisors are Professor Connell and Dr Dominic McAfee. And so today I have the pleasure of sharing with you my research proposal, and I'll start by giving you some background information into this topic, and then I'll go into my aims and objectives, my methods, and the significance of this research. So firstly, for some background information. Well, worldwide, we have lost and are continuing to lose our marine habitats, and this is due to things like overfishing and habitat degradation, as I'm sure you all know. And so many complex, healthy habitats like coral reefs and oyster reefs are losing their complexity. So they're becoming bare, sandy kind of habitats that have little biological life, little productivity, and little biological noise. So essentially, they're barrenscapes. And this loss is so pronounced that 2021 to 2030 has actually been declared the decade on ecosystem restoration by the United Nations. And so many large-scale restoration projects have now emerged to try and help this. But if these are to be successful, we actually still need more scientific knowledge on the best restoration techniques to use. And so the research that I'm focused on is helping Australia's native flat oyster reefs recover. And so these oysters are called Ostrea and Gassi. And just 200 years ago, these reefs extended over 1,500 kilometres of South Australia's coastline and provided with them many ecosystem services. But now we only have one native population left, and that is in Georges Bay, Tasmania, as they've been indiscriminately removed from the mainland, predominantly due to overharvesting and dredging. And so, as they haven't naturally recovered themselves, it's up to our restoration efforts to try and restore them. And so, soundscape enrichment is a novel restoration idea, and it could just have the potential to help safeguard these restoration projects against failure. But what is a soundscape anyway, and how can EnrichNet help restore these oyster reefs? Well, firstly, I'll give you a bit of background into sound in the marine environment. So the underwater world has long been viewed as a dark, deep and silent realm. And this is largely due to the fact that the ocean is much harder to access than its terrestrial counterpart is. But there did arise a shift in this widely accepted opinion during World War I, with the rise in enemy submarine activity. So war at sea meant that acoustic sonar, hydrodynamics and echolocation each had to be used to detect torpedoes and improve submersible technology. And with this rise in underwater warfare technology, there was also the discovery of a strange noise in the sea. And this came in the form of a background crackling noise, and it interfered with sonar performance. So naturally, the US Navy thought that it must be jamming by the enemy. Now, other guesses as to its source were that it was the expansion and contraction of the ship's hull, rolling pebbles, static crashes, maybe shipworms eating away at the timber, and some guesses were even that it was the clapping away of oyster and clam shells. But eventually, it was discovered that the culprit of this noise was the snapping shrimp. So the snapping shrimp is a marine invertebrate with a large snapping claw. And when it closes its claw, it produces and collapses a cavitation bubble. And at source point, the noise from this collapse can exceed 180 decibels. And so a single snap can make a loud snap or pop kind of sound. Whereas if you have a whole population of these snapping shrimp, it produces this crackling noise. And it sounds a lot like bacon frying away on a fry pan. And interestingly, with this discovery, the Navy even started to use snapping shrimp as a weapon. So they would start to hide their submarines in snapping shrimp beds because the cacophony of their noise masked the enemies using sonar. And so with this discovery all these decades ago, our oceans began to be viewed as a diverse, vibrant and cacophonous world, rather than this undisturbed, silent one. And so the underwater world, it could potentially even be viewed as an orchestra. And so you have your snapping shrimp snapping, you have whales, you have fish drumming and honking, you have whales clicking and dolphin whistles, and you have a myriad of other sounds produced by other animals. And so along with ambient noise, wind, rain, waves, and noise from shipping, these collectively produce what we call the marine soundscape. And so apart from being musical entertainment for the Little Mermaid, like seen in the movie with Under the Sea, why are these sounds so important? Well, they could actually have great implications for our restoration and conservation efforts. So back in 1983, a terrestrial researcher named Kress actually found that using playback of bird sounds could help re-establish bird populations. And so he was the first study that I could find that suggests using playback of sound for restoration and conservation purposes. And so he used little models of Arctic tern birds and laid them around his site and had playback of their calls. And within 3 years, he got them to settle again in this area and to nest and reproduce. And since then, a few other studies have looked at sound for restoration. These predominantly come in from bird and frog studies. And then a few have also come from fish and shellfish studies. And so our figure on the right here pretty much shows the effect size seen between playing sound to animals compared to no sound controls. And for each of these animal groups, you are seeing a significant effect size, which would indicate that sound does have an important role to play. And so you could even say that the underwater world might have highways of sound that recruiting organisms can detect from afar and follow all the way home. Because sound in the marine environment travels really far because it experiences little sound loss. And it also is present at all depths in the water column, and it can still travel regardless of whether or not there are currents. And so arguably, underwater sound is the best cue that organisms could use as a long distance navigational cue. And compared to cues like visual and olfactory ones, it travels much further. And so this here is a figure on the spatial scale that different sensory cues operate at in the marine environment. So at the end here, we have the homes of the fish, which are several kilometers away from their starting point. And so our fish at the top uses sight alone, so vision, to try and find home. But even under the best conditions, most organisms are unlikely to see more than 50 meters away. And so this fish did not find home. Whereas the one below it, he used olfactory cues, so smell. And his cues were traveling with a current, and so he did get quite far. But eventually the current broke down and his smell dissipated, and so he was once again lost. Whereas our fish on the bottom used these auditory cues, and he travelled along his highway of sound and made it all the way home because his cue wasn't affected by light or currents or anything like that. But what if these highways of sound disappear? How do recruiting organisms find home? And how do we ensure we keep getting recruitment to reefs we are trying to restore? So habitat degradation in the marine environment is turning these loud, healthy, complex habitats into bare, sandy, quiet ones. And so during my honours program last year, I looked at habitats of different complexity and recorded their soundscapes. And I found that as habitats get less complex, they get quieter. And so these are some spectrograms I produced, which are just a visual way of seeing the sound in the landscape. And so the red and orange colours indicate loud noise, and these ones come from snapping shrimp in about the 2 to 5 kilohertz frequency range, which is on the y-axis there. And so rocky reef on the bottom is the most complex habitat that I recorded. And as you move upwards towards the sand, you're decreasing your complexity in habitat. And we can also see that as they decrease in complexity, they get quieter as well. And I also looked at snapping shrimp snaps per minute in each habitat, which is the figure over here on the left. And so we're once again seeing this gradient where the more complex habitats have more snapping shrimp snaps, and you're getting fewer and fewer as you head towards the sand, which makes sense because you're not going to have many loud noise producers in a bare sandy habitat. And another metric was the sound pressure level, and you're once again seeing this kind of trend. And just to help you understand this a little bit more, I've got some recordings that I took last year. So the first one is of sand habitat, so you won't hear much because it's very quiet. Might hear a little crackle, but that's about it. Whereas the next one is from rocky reef. So you're hearing that crackling background noise from the snapping shrimp. And so this is where our idea of soundscape enrichment comes in. So essentially the idea is that we'd place underwater speakers into bare habitats that are recruitment shadows in that they don't have many cues that can come and attract the organisms to them. So we're placing that cue there and playing healthy habitat noise, and we'd hope that we can attract baby oysters to come and settle in this area. But in time, we would hope that we'd be able to remove the speakers because the reef would be able to produce this noise themselves. So when we get the oysters to settle, it should encourage fish and invertebrates and all those noise producers to come back and create the noise on its own again. And so we have the theory behind this idea, but we're still lacking that scientific evidence. And so the research gaps I'd like to fill are, firstly, is soundscape enhancement a feasible habitat restoration technique? Which marine soundscapes can actually provide the best cues to attract and induce the Oangasi larvae to settle at restoration sites? And if the larvae is attracted to certain soundscapes, What are the thresholds of the sounds? What spatial scale does it operate at? And what mechanism do they use to detect sounds? And lastly, if it's a feasible tool, how can we implement it effectively in our restoration practices? And so my research aims are to firstly gather the practical scientific knowledge on novel restoration strategies to improve large-scale restoration efforts and try to safeguard them from failure. Secondly, to develop new strategies to improve the effectiveness of oyster reef restoration projects in Australia, pioneer new methods that could also potentially be applied worldwide. And lastly, to develop underwater speaker technology to determine the potential of enriching a key settlement queue, which is the marine soundscape that larval oysters use to locate settlement sites, where restoration projects are limited by recruitment shadows. And so to do this, I've begun work with an NGO, and we're producing affordable, easy to use marine speaker technology that should hopefully help to improve landscape scale restoration. And so these are really simple designs, so it's pretty much got everything you need to play the sound within this PVC unit, and you seal it up and waterproof it, and it's much, much cheaper than those currently available on the market. And so we'll do tests with these and hopefully use them throughout my project and maybe restoration projects in general. And so I've just got a little recording here of this speaker playing this snap and shrimp noise, and it's very loud, so just mind your ears. Yeah, and I will also like to assess which marine soundscapes are the best cues for attracting and inducing settlement of these baby oysters. And so I will use a collection of the sounds from my honours program, these being the rocky reef, oyster reefs, seagrass and sand, along with other sounds like white noise, some snippets of sound from sons, and then no noise, just to see which kind of sounds the oysters seem to like. And I'll do this in both the lab and the field. And then I will see whether enriching these sounds that they prefer will actually boost their recruitment. And whilst doing this, I'll try to figure out the spatial scale that the oysters can detect this sound at, as well as which sensory structures or mechanisms they may use to detect the sounds. And lastly, I'd like to continue to build my collection of soundscapes from oyster reefs to include reefs of different age, size, and functional health. And this should help us establish a threshold at which we may no longer need to use these underwater speakers, so help us establish a healthy sound level for oyster reefs. And as we've already taken recordings from those in South Australia at our restoration reefs, these will come from New South Wales and Tasmania. And so if we can attract more oyster larvae to restoration sites, then this project should be the first to increase oyster recruitment to a restoration project, which is a significant advance in ecological restoration. And it's particularly important because at the moment there hasn't been much practical application of soundscape theory to actual restoration efforts. So it should provide a new and affordable strategy to help improve landscape scale restoration and safeguard these efforts from failure. And thank you for listening. If you have any questions, feel free to type them into the chat or to ask me if you unmute. Alternatively, you are welcome to email me. I've got my email on the bottom right here. And one last thing, if you would like to hear more sounds from the Oceans Orchestra, I'd recommend this link on the bottom left here. They've got a really big, nice collection. So, thank you.
Stuff.
Might just stop share so I can actually see the chat. So Diego has asked a question. He says, do the snap-in shrimp use their snaps for communication? And if so, is this the reason why they get louder in complex environments? So far we think that snapping shrimp make these snapping sounds either to communicate with one another or perhaps during aggressive agonistic signaling behavior and things like that. So if like a predator's come in, they might snap to indicate that to others, or just to snap to communicate with one another. But we think the reason why snapping shrimp noise is louder in more complex environments is because these complex environments actually have more places for the snapping shrimp to live in. So more structure, more rocks and shell and hidey holes, because then they can hide there from predators. If it was just a sandy habitat with nothing to hide in, they'd be easy game for predators.
I have a question, can I?
Yep.
Hi, my name is Tim from OIST.
Hello, Tim.
Hi. So very interesting project. So my question is, do you know how far the sound is actually propagating in the water?
So I don't have any numbers on it yet, but shortly I'll be doing some tests to look at that with the sound recordings I'll be using. So there's things called hydrophones, which are underwater recording devices. So the plan will be to place underwater speakers in the water, playing my sound, and have a gradient of these hydrophones. And then each hydrophone will pick up the sound, and I'll produce those spectrograms like I showed before. And then you'll be able to see how far the sound travels and whether its quality stays the same and decreases and things like that.
Yeah, the other question I have, do you talk about the impact of this sound on other marine life?
Yeah, so at the moment, obviously my focus will be on the oysters, but there was a study by Gordon at the end of last year, 2019, and he was using coral reef sounds up at the Great Barrier Reef, and he was broadcasting those sounds, and it attracted lots of fish, both juveniles and adults. And so, yeah, that's one study that shows it for fish. And then, yeah, so for some organisms, these sounds seem to be positive, but I guess for lots of other organisms, perhaps they'd be negative. We don't know yet. So, yeah, it's a big research area that has lots of gaps.
Okay, thank you.
Yeah.
Very pretty cool project. Thank you.
Linda asks, if the snapping shrimp is possibly aggressive behavior, could it deter other organisms whilst attracting the oysters? Potentially it could. I haven't really thought about that, but that would be something to test as well, I guess. Another branch of my research I was thinking about would be to actually attach underwater cameras or light traps with these speakers when I lower them down to see what kind of organisms are coming and which ones aren't hanging around. And yeah, that might be something that gives light to that question. And Jasmine asks, exciting research, Brittany, how are your suitable shadow habitats identified? As in, how are you assessing habitat quality? So essentially, I didn't have any numerical values or anything. It was just going out and looking at the habitats myself. And so just seeing which ones were predominantly sand or seagrass or had this reef structure. Okay, and I think that's all we've got at the moment, unless anyone has more.
Following up from that, if you think, Brittany, that the snapping might actually bring other shrimps in, and eventually you could take your microphone or your speakers away, and just the whole system would actually just get the ball rolling. Maybe that's the role of this beginning by playing the sounds there. Could take it away by the other animals that come along to that.
Yeah, that's right. So I think our idea is to put the speakers down kind of as a way to boost the recruitment of organisms at the start and to get those sound levels up and going again. And then we'd hope that we wouldn't have to keep the speakers there and we could remove them once they've reached a healthy threshold again.
Great. Well, thank you so much, Brittany, for sharing your exciting research. I love your little soundscapes and the fact that you could share them, and just the absolutely innovation that you're bringing to this space. So exciting research, and we really look forward to hearing more down the track. So everyone, please put your hands together for Brittany. Thank you very much. Great. And then I'll screen share again. I'd like to introduce our second speaker, Vittoria. So Vittoria is passionate about our oceans and their marine life, but she's also concerned about the threat of climate change to them. So Vittoria's PhD is focusing on fish population dynamics under future climate scenarios. So without further ado, I'd like to hand over to Vittoria to tell us about her research.
My research in particular focused on the effects of climate change on fish reproduction. So I know that many of you already know what is climate change, but I just want to give you some more background information. So climate change is a change in the pattern of weather and related changes in oceans, land surface, ice sheets, and it's occurring over a long time scale, so over decades or longer. Climate change can drive changes in our atmosphere, and these changes can cause effects on oceans, such as warming, temperature, while ocean acidification is directly also caused by an increase in carbon dioxide in our atmosphere, caused by the burning fossil fuels to power houses, cars. So human activities are increasing the emissions of the atmospheric carbon dioxide, driving rapid changes in the physics and chemistry of the environment, as never have been recorded before. So this is a video of 10 years of sea surface temperature changing. So ocean systems act as a heat sink, and to date have absorbed approximately 1/3 of the human-produced carbon dioxide from the atmosphere, with a resulting warming and acidification of water masses. Therefore, physical and chemical changes in the marine environment can impact biological functions of marine organisms, initiating a wide range of responses which have profound implications at individual level and to the dynamic of the entire ecosystems. And ocean acidification might not be the first thing that comes to your mind when you think about the impact of carbon dioxide in the atmosphere, but currently ocean absorbs about 1/4 of the carbon dioxide in the atmosphere. And what happens when all the gas of the atmosphere interact with seawater, and carbon dioxide mixes with the water, and it forms carbonic acid. And the increase of carbonic acid leads to a depletion of carbonate ions, which are essential component of minerals, which are vital, for example, for building seashells. And acidity instead is measured on a pH scale, where 0 is most acid, 7 is neutral, and 14 is most basic. And since the Industrial Revolution, the ocean pH has dropped from 8.2 to approximately 8.1. And while this does not sound like much, that is actually a 30% increase in acidity, because it's a logarithmic scale, so each digit on the scale indicates a change by a factor of 10. So a pH of 7 is actually 10 times more acidic than a 8. So at current emission rate, ocean pH might drop to a 7.8 by the end of the century, which is going to create an ocean which is more acidic than any seen in the past 100 million of years. So under such increasing pressure and habitat loss, organism needs to adapt, move, or die, relocating to new habitats with more favorable conditions for their survival. So migrant species can trigger new interactions and shifts in their novel ranges. So this can have potential consequence in the community function, disrupting the ecology in the newly occupied system. And global stressor challenge also the tolerance of organisms with costly physiological adjustments. For example, ocean warming increases metabolic rates and respiratory demand, whereas ocean acidification increase the energy expenditure towards homeostasis processes. So life-supporting activities are normally prioritized over the development, movements, somatic and gonadal production, and storage as well, because the basal maintenance is the priority for the survival, and then organism will allocate the remaining energy to the other fitness-related activities. But first is the basal maintenance. Therefore, an increase in temperature and carbon dioxide can directly and indirectly drive behavioral and physiological alteration at organism level, which are then affecting the fitness and species performances, which in turn can have consequences for species interaction and key ecological processes, which then they can translate to responses at community level. Reproduction is one of these key ecological processes because it sustains the individual fitness and population replenishment. Reduced energy availability for fish could potentially reduce the clutch size. They could produce smaller eggs, smaller hatchling, or even fewer clutches produced in a breeding season. And how so reproduction is affected given the rising energy demand driven by this global stressor and resources availability. Donaldson found that the spiny chromis reproductive output decreased at elevated temperature and stopped completely when it was combined with limited food availability. So in this experiment, they had 3 different levels of temperature with 2 food regime. So they have an average food consumption and a minimum feeding rate. So when normally we have a pattern of decrease with increased temperature, when together with the food availability was decreased, the reproduction completely stopped. On the other hand, an elevated input of carbon dioxide has shown contrasting effects even among species of the same family. So for example, if I can do this, and in this case, in the study of Miller, they found that the reproduction is actually stimulated the production of clutches in a species of the cinnamon anemone fish. So there was a stimuli in the reproduction with a high level of carbon dioxide. Contrasting, Welch and Monday in the second study found that in the tropical species of damselfish, although they were reared under the same elevated condition of carbon dioxide, had contrasting responses. In fact, there was an increase and decrease in the reproductive output respectively. So the anemone fish had a higher level of eggs produced compared to the spiny chromis. So when we put all of this together, so temperature and carbon dioxide, there's only one study from Miller. It was a multi-stressor study, so it found that the potential stimulatory effects increased by high carbon dioxide can be reversed by the elevated temperature, and the final output will be negative. So they use this a long-term study with 3 different level of carbon dioxide and 3 different temperature. And as we can see from the results, they recorded each egg clutch produced during the breeding season, the number of eggs laid per clutch, and average egg size, fertilization success, the survival of the eggs as well. So all information about the reproductive output. And temperature had by far the strongest influence on reproduction. So there was a decrease in this part, a decrease in the reproduction. And the carbon dioxide instead had a minimal effect in the majority of the reproductive traits that were measured. Instead, when they were coupled together, because here we can see that by himself in the control and with a moderate carbon dioxide, the number of clutch actually could increase because this is the potential stimulatory effect. When together with the highest temperature level, also if there was a stimulatory increase in moderate temperature, when at the highest temperature, the potential stimulatory effect ceased completely. So the final output was negative. So our current knowledge on fish reproduction under climate change pressure is limited. And moreover, a laboratory experiment which considers stressor in isolation do not take into account other influencing factors, such as the food supply, predatory prey interaction, and competition. So we have a limited understanding on fish response to future climate conditions. Energy reallocation also towards primary vital activities and reduction in resources availability are predicted to drive a decrease in reproduction investment. Hence, we still have a lack of understanding about the potential stimulatory effects of these changing environmental conditions on fish reproduction and the eventual reversal effects, specifically how the interplay of ocean acidification and warming can affect the fish fitness in terms of reproductive behavior and performances. Therefore, my research aims is to unveil those effects and understand how ocean acidification affects reproduction from different aspects. So I will study the natural behavior in the wild of fish, and physiology of the adult and their eggs, and the sperm competition using genetic tools. And lastly, to predict the impact of both ocean acidification and increasing temperature on the energy budget of the whole organism, and thus predict through modeling the possible outcomes on the dynamic of their population. And how I will do this? I will try to answer all these questions using volcanic vents in Japan as a natural laboratory of ocean acidification, focusing on species with male parental care. So for the behavioral observation, I will use video recordings without the presence of a diver, so with using a GoPro in quadrants. I will use the vent size on a control site to compare, and then analyze their behavior, which the one related to reproduction. So in particular, this is a video of how it is a male guarding his nest. So nest guarding behavior, so how much time they will spend inside the nest, if they aerate the eggs, they remove the drift material, how they defend from predators, their feeding rate, and also if it's possible to observe a courtship behavior if the territorial male is trying to attract females. For the physiological analysis, I will use the male, collect males and eggs with their nest, so use the male total length and body weight to establish the relationship between, if there is a relationship between size of the adults and also number of eggs per nest, so for how many eggs they can take care of. And also we'll extract the liver and gonads to obtain an estimate of the energy investment for reproduction and for storage. And the body condition would be important to examine any potential physiological mechanism for changes in reproduction. And we will use a picture of the nest and we will use a software to quantify the number of eggs per nest and their size. And also we will use lipid analysis to quantify the maternal investment in the eggs with fatty acid profile. Concerning genetic analysis, I will do paternity test, collecting the DNA of dominant males and the eggs. I will extract and amplify with PCR using microsatellite developed for the species. And once obtained the genotypes for adults and eggs, we will assign the paternity to eggs based on strict exclusion. And then I will be able to analyze the results. As an example of paternity test, I will be able to analyze the result on analyzing the pics on the computer, and so we will be able to see with genetic tools what we cannot actually see on the field through only behavioral observation, because genetics can tell us more than what we cannot see. And so if there is any sperm competition given by sneakers that try to fertilize eggs not of their nest. And lastly, the DEB model, so dynamic energy budget, is a model that will be used in this study to predict how ocean warming and acidification together could alter the fitness performance of a generalist fish species, the triple fin. This mechanistic model provides a theoretical context to understand how energy is allocated to different physiological processes for organism survival. Importantly, the model accounts for environmental factors such as the food availability, the temperature, and I will introduce the variable of ocean acidification and how this variable influence the organism physiological processes, and in particular, growth and reproduction. The novelty of the proposed research is in the study of the species in their natural environment, in which interaction with other organisms can shape their behavioral responses. Using natural laboratories of ocean acidification, it will be possible to have insights in the end of century climatic condition and see how such stressors have affect the reproductive output in organisms which have been exposed all their life to high level of carbon dioxide, and this has never been done before. And as reproductive output is extremely important because it's intrinsically related to population replenishment, it's important to be able to predict the species behavior and adaptation. And this, with such a comprehensive approach from behavioral, physiological, and genetic point of view, hopefully we'll be able to answer all these questions. And this brings me at the end of my presentation. I would like to conclude with a nice comic and stressing that understanding the basis of this variation will be critical for predicting the fate of reef fish population and community in future high carbon dioxide world. Do you have any questions for me? Thank you. I don't see any. So I stop sharing my screen. Open the chat. You have any questions?
Hi, Vittoria. Tim.
Hi.
I don't have a question, but I have a suggestion. When you're talking about the macro satellite analysis,
Yes.
I think you should more consider right now rat sequencing, which is a more general and faster technique than and more accurate than macro satellites. So,
Which one? The
rat sequencing.
Yes.
RAD sequencing. Restriction enzyme sequencing. So it's more global. So you basically you cover a lot of macro satellite at the same time. And in terms of price and support is more or less, well, a little bit more expensive, but not too much. But you can cover, you know, a deeper field. So just a suggestion.
Thank you.
But great presentation. Thank you.
Thank you.
So Vittoria, I got a question for you. So what's your, while others are waiting, what's your prediction in terms of the paternity analysis? So what's your a priori expectation how ocean acidification might alter paternity?
Because for what we know now, which was this study from the vents in Italy, they found that it's not really altered the paternity because the ocean acidification can change the behavior. So actually, if the father can take care better of his eggs, he will be able to stop other males to fertilize his eggs. So
So you're predicting super dads in the future?
Depends on the species also, because not all species have sneakers. So not all species have sneakers where they don't have a proper nest and they go to fertilize other male nests. So I think that only with ocean acidification, they will not have a negative impact. But it's my personal opinion. Because at the moment we have a stimulatory effect, so it's positive. So I am positive also for the genetic part. But we will know later. Okay. Good idea.
Sorry, we can't hear you, Sean.
Yeah, I'm sorry, I didn't want to talk over the top of you. Victoria, yes. In one of your graphs, you showed that the clownfish were improving their reproductive potential as the waters became increasingly acidic.
Yes.
Do you know why that would be?
Only in the carbon dioxide, right?
Correct.
So they consider as all of it potential, so they use the clutch laid per pair of clownfish and the number of eggs, the eggs area, and so this all of it was the reproductive output. So you ask me why or what?
Yes, I was wondering if is there any speculation on the mechanism?
There are studies that are trying to, this is about transgenerational plasticity, so how if they're able to pass also this information, so this improvement to the following generation, that there are no studies at the moment about that, but this is just how they adapt. So that's how the parents, so if it's a short-term adaptation, they're able to produce more eggs. Mm-hmm. But we don't know about long-term if they will be able to sustain these changes, because it requires a lot of energy investment in their eggs. So in short term, they are able to invest enough energy, even if they have to spend energy for the acid-base exchange, so for homeostasis. So they're still able to have enough energy for the reproduction. But this is for the short term, not long term. So we don't have an answer for this other case.
Okay. Well, thank you very much. Sounds like a very interesting project.
Thank you. And I have a question from Jasmin. Is it possible to assess differences between individual and population fitness in your fish? In terrestrial species, this can help to improve prediction on population viability. Is this dual approach possible with fish to assist your prediction for population dynamics under future climate scenarios? So what I'm doing for the population point of view, I'm doing with the modeling. So the model helps me, I will assess first the energy budget of a single individual fish, and then this will give me, I will be able to predict about the population. So, okay. Yeah, so this is what we're trying to achieve with the DB model. Starting from just the physiology of individual fish to obtain a prediction for the whole population. Any other question?
Great. Well, thank you, Vittoria.
Thank you.
Please put your hands together for Vittoria.
Thank you.
Thank you so much. Great. Now, I'm not sure how to take the spotlight off you. So you're in the spotlight for a while there, Vittoria. So thank you to Brittany, thank you to Vittoria for 2 excellent seminar presentations today. And let me just share of exciting of what we've got coming up next. So we now are having the seminar series as the first Friday of the month.
Sorry, everyone, I have to leave now. Good to see you all. Nice to work, Vittoria. Sean, good to see you.
See you, Tim.
Great to see you.
See you soon. Sorry, guys, for another Zoom call. Bye.
Thank you.
See you, Tim.
Bye. Great
Bye.
to see you.
Thank
Bye-bye.
you.
Thanks.
Supervisor logging off there. It's great that he could zoom in. Fantastic. So yes, these seminar series are presently the first Friday of the month for our winter seminar series. Our next up is on the 3rd of July. And we're really excited to have Mickey Wang joining us from Bertram's lab. So Mickey is going to be bringing to us new technology to detect viral diseases in vineyards. So all you wine drinkers out there, fellow wine drinkers out there, it's very important research that we're going to be hearing from Mickey. And we'll also have Professor Megan Lewis sharing and introducing how our spatial sciences research group are matching exciting remote sensing with environmental assessment and monitoring needs. So hearing from that spatial sciences approach. So we hope you can join us next month, 3rd of July, to hear from Mickey and also from Megan.